Hello, good morning, everyone. I'm Ivy Chung from Pharmacology Department. And today it is really a great pleasure for me to introduce a collaborator of mine, Dr. Yolanda Augustin. She's a Malaysian clinical oncologist based in UK, but she has a special interest in developing affordable diagnostics and therapeutics for global oncology and also sarcoma service in low and middle income countries. So she did her undergraduate medical and also palliative care training at Guy's, King's, and St. Thomas School of Medicine in the UK before she undertook clinical oncology training at the Royal Marsden, London. After that, she took a research fellowship with the Royal Marsden Sarcoma Unit, and that's when she got into more training in clinical trial design and also delivery for novel systemic anti-cancer therapies. So with her extensive clinical trial portfolio, she's now working with different colleagues, especially from Malaysia, on developing more novel therapeutics and also diagnostics under the oncology. So currently, she's just recently been appointed as a visiting fellows academics in part of Faculty of Medicine, and I think this is a good platform to introduce her work and also to our colleagues here in faculty. We need to promote what we are doing here and as well as to encourage more collaboration. So with that, I would like to invite Dr. Yolanda to take on the stage.
No, thank you so much, Prof. Ivy, and selamat pagi semua. Great to see you this morning. So today I will take you through a whistle-stop tour of drug repurposing for Malaysia and ASEAN and how we can actually go from bench to bedside. But a lot of the content I think might require more in-depth discussion. So after this meeting, please feel free to reach out to me. We can discuss in more detail. And I would like to start by saying thank you very much to Prof. Ivy Chung, who's become a very close collaborator and friend over the last couple of years as we've been working together to build up this program for drug repurposing at and also Malaysia, and also to my mentor and former PhD supervisor, Professor Sanjeev Krishna, who's an infectious disease specialist who has a special interest in one of the drugs that I'm going to discuss today, which incidentally is also an anti-malarial drug. He's sorry that he can't make it this morning, but we would love to also meet with the rest of you if everyone's interested on anything that I mentioned in this presentation when he's back out in Malaysia in October. So this program that we're discussing is actually part of a bigger project called the International Affordable Diagnostics and Therapeutics Alliance. So this alliance has just been newly launched in Malaysia as of January this year, and it brings together Ministry of Higher Education with Ministry of Health to work on three main pillars. The first one is about affordable diagnostics, and for that we have a few projects that are in place, but they're focusing more on point-of-care diagnostics for areas like Sabah, Sarawak, and rural settings. And then the second project we had is about drug repurposing, and that's how we can actually make Malaysia a center for drug repurposing in ASEAN and also the Global South. Initially with cancer, but we're also looking at other potential disease areas, including rare diseases and a few others that I'll mention at the end of the presentation. And the longer term project, which is in collaboration with SIBA, is about plant molecular farming. So that's where we can actually genetically modify plants to produce cancer immunotherapy or cancer antibodies, monoclonal antibodies, and therefore bring the cost of treatment down. So the reason why we wanted to set up IA Data in Malaysia is because we saw an opportunity to bring together strong intersectoral partnerships with government, academia, responsible industry, and civil society to really look at therapeutic solutions and also diagnostics for neglected diseases, cancer, and rare diseases in the Global South. And we saw a very unique opportunity in Malaysia where there's a lot of capacity within the academic research setting. But we wanted to help create more scientific exchange and knowledge exchange collaborations, both within Malaysia but externally as well, across ASEAN and internationally. We also wanted to help Malaysia to attract international grant funding and foreign direct investment into Malaysia. And I think this is quite timely because now under Malaysia's NIMP 2030, the new Industrial Master Plan 2030, pharmaceuticals and medical devices are 2 out of 5 key sectors that the government is focusing on. And how the collaboration works is that we have a lead entity at University Malaya called the Affordable Diagnostics and Therapeutics Research Group, which is led by Professor Ivy. And then myself and Prof. Krishna, although we are academics at City St. George's, University of London, we also are leading a social enterprise called Cadet. So that's the Centre for Affordable Diagnostics and Therapeutics, which is registered in the UK. But the aim is to be a sort of matchmaking entity where we help to bring in international connections, both on the academic side, but potentially in the future on the philanthropic side to support some of this work in Malaysia. And then we also work very closely with Clinical Research Malaysia under Ministry of Health to essentially help with our organizational setup of IA data and also connecting with NIH, the National Institute of Health, be it through IMR and ICR. And then we also have a Third World Network and the South-South Diagnostic Alliance, which are part of our senior management committee and part of the organizers of this alliance. In Malaysia, at the moment, we are connecting mostly with and UNIMAS, but in the future, we'll also be looking to expand to some of the other universities in Malaysia. And then we also have a few entities that we're bringing in from ASEAN, which includes PESA, which are our colleagues that are primarily based in Vietnam. And we also connect with the NGOs or the CSOs on the ground that are particularly interested in this cancer space, such as NCSM and SCAN in Sarawak. And then we have a few industry partners that we are also working with. But for the purposes of today's talk, our main industry partner is Duo Pharma because they are a government-linked company and generic drugs manufacturer. And then at the international side, we are also working together with some international entities on the diagnostics and therapeutics side. And we basically bring them in on a project-by-project basis. So what is drug repurposing? So when I first started to work on drug repurposing, like nearly 10 years ago now, it was considered a bit fringe. And I think not a lot of interest really in this area in a way, because it was seen as the domain of mostly academics. And to actually get pharmaceutical companies or industry to be interested in drug repurposing, where often they do not see a huge profit margin, was a bit challenging. But I think we were actually helped by the COVID-19 pandemic because this really accelerated interest in drug repurposing. And for example, in the UK, where there was a big drug repurposing study run by collaborators and colleagues at Oxford called the Recovery Trial, they actually looked at repurposing 16 different generic drugs for COVID-19. And actually 4 out of the 16 did come to a clinical indication. So I think that actually helped to open up the drug repurposing space and make everybody more aware that this is one potential way that we can accelerate drug development. And if we listen to the sort of traditional mantra around pharmaceutical development, we're told that it can take up to 15 years to go from bench to bedside at a cost of up to $1 billion to actually deliver a new drug into the clinic. And drug repurposing, I think, does give us an opportunity to try to short circuit and technologically leapfrog this long process and pathway. So for a number of years now, we've also been working with Pan Panziaka, Dr. Pan, who is a bioinformatics specialist who also has a big passion for drug repurposing, particularly in the sarcoma space. He has been working together with the Anti-Cancer Fund in Belgium, but they've now also become a part of this big project in the EU called Remedy for All. Which is funded by the EU Horizon grant of about 23 million euros to basically set up this platform for drug repurposing for Europe. And on it, they have some very good training modules. They run a conference every year. They also connect with industry and academics to look at how to do intelligent clinical trial design to really try to get some of these repurposed drugs into the clinic. So we're embarking on a strategic collaboration with them between Malaysia and Remedy for All, where will be the lead entity from the academic side. And we're hoping that in the next few months, we'll be able to plan some very interesting workshops, knowledge exchange, and then towards next year, we're also looking at hosting a conference in Malaysia together with Remedy for All, where we can really help to make repurposing more mainstream and bring in more interest across the faculty in but also across other academic institutions in Malaysia, and also hopefully Ministry of Health. So on the Anti-Cancer Fund and the Redo database, there's basically some very nice work that's been done looking at the bioinformatics aspects of drug repurposing for cancer. And to date, there are actually more than 300 generic drugs which are off patent, relatively affordable, and generally with good safety profiles, which actually potentially have some anti-cancer activity or benefit. A lot of these studies, though, although there's a lot of preclinical data and lab studies or lab papers looking at the anti-cancer effects, there is a challenge in terms of taking these forward into clinical trials and therefore eventually into the patient setting. And if we look at the drug repurposing area, there's only actually been about a couple of repurposed drugs between the 1990s and 2021 that have actually made it into repurposing for cancer. One of them is rapamycin for renal cell cancer, and the other is raloxifene for breast cancer in certain high-risk postmenopausal women that have been licensed for a new therapeutic indication for cancer. Historically, there have been some successful examples of repurposing in oncology, including thalidomide for multiple myeloma, ATRA and arsenic trioxide for APML in the leukemic setting. And then interestingly, a few years ago, the CAP2 study in the UK actually managed to show that there was a benefit of aspirin chemoprophylaxis in hereditary colorectal cancer, Lynch syndrome. So that's actually part of NICE guidelines now as well. So how might these drugs work? So really trying to understand the mechanism of action to link biological plausibility is very important. And right now we're trying to work out what are the clinical settings in which some of the agents that we're interested in might actually have a benefit. So this includes whether or not they might work in the precancerous setting. So some of our work is looking at precancerous conditions. Also, whether there might be effects in terms of the tumor microenvironment and microscopic metastasis, so targeting the neoadjuvant setting. Looking at combination therapy, so are there ways that repurposed drugs can actually help to make chemotherapy or immunotherapy more effective? Or could they also be a radiosensitizer when used in combination with radiation therapy? And of course, there's a lot of interest as well in terms of drug repurposing in the adjuvant setting. So what happens to patients after they complete their primary oncological treatment if we don't have any other proven treatments to give them after their primary surgery or chemoradiotherapy? Could there be a role for drug repurposing in preventing metastasis and recurrence after primary oncological treatment? So this has led us over the last 10 years to focus on one particular drug called artesunate, which is an anti-malarial. And that's where Professor Sanjeev Krishna's expertise come in. And that's where I joined him as a PhD student towards the end of my clinical oncology training to basically work on drug repurposing for cancer, looking specifically at artesunate for colorectal cancer. And we've now set up at the artesunate repurposing drug mechanism and novel trials for the Global South, Ardent GS Consortium. So artesunate is actually a very interesting drug. It comes from a herb that's been used in Chinese traditional medicine for thousands of years called Qinghaozu. And if you look back at the traditional Chinese texts, it says to travelers that if you are traveling, keep the tea leaves up your sleeve. And if you have a fever, then brew the leaves. So potentially they might have been treating malaria, I guess, 2 millennia ago. But it was actually discovered by a Chinese scientist called Prof. Tu Yu back in 1972. And back in 2015, you might remember that she was the first female Chinese scientist to be awarded the Nobel Prize for Medicine. And it has been used in hundreds of millions of doses as a safe and affordable anti-malarial drug globally. So at we are looking at some specific areas at the moment, which are to do with colorectal cancer and cervical cancer. We have a clinical study, which I'll discuss in a minute, which is a phase 2 study for colorectal cancer and also a phase 2 study for pre-cervical cancer, CIN 2 or 3. But in the future, we'd also like to look at IBD, inflammatory bowel disease, as the precursor to colorectal cancer in some cases. And then our collaborators at CMC Vellore, Christian Medical College in Vellore, are also working on acute myeloid leukemia. And over the last 10 years, they have done some very nice mechanism of action work in collaboration with our group at St. George's. And this has now led to a phase 1 study in CMC Vellore of artesunate in combination therapy for acute myeloid leukemia. And we're also just starting mechanism of action work together with Prof. Ian Patterson and Prof. Lee Fa on nasopharyngeal cancer at So how might this drug work? Well, if you look in the literature, there are thousands of publications discussing the potential anti-cancer mechanisms of artesunate. And basically, broadly speaking, it does affect multiple points in terms of anti-cancer properties, including blocking cancer cell proliferation, promoting cancer cell death through various mechanisms, blocking critical cancer signaling pathways, and also invasion, metastasis, and angiogenesis. But also, very interestingly, it appears to attenuate inflammatory pathways in multiple models of inflammation, not just in cancer models. So we're also hypothesizing that this is probably the common mechanism of action that artesunate may have a benefit for, particularly in the precancerous lesions, where inflammation plays a very important role. And we're also looking at how we can develop artesunate as an anti-metastatic agent based on a framework that's been developed by Cancer Research UK and also the Australia Metastasis Working Group. And they've come up with this very nice publication that has really sort of changed thinking about the ways that we could actually look at repurposed drugs as anti-metastatic agents. And I encourage you to read it if you haven't come across it before. So this is just the chemical structure of artemisinin. I won't go into too much detail, but just to say that one of the fundamental ways that artemisinin is, or artesunate is thought to work in cancer is actually through the endoperoxide bridge. And artesunate, which is a derivative of artemisinin, is characterized by a crucial endoperoxide bridge in its chemical structure. And in cancer cells, the endoperoxide bridge is cleaved in the presence of ferrous ion, Fe2+, which is often abundant in the tumor microenvironment. And this cleavage leads to the generation of highly reactive oxygen species, including free radicals, which can cause oxidative damage to cellular components such as DNA, proteins, and lipids. And what's also interesting is that there is a selectivity for cancer cells because cancer cells often have a higher labile ion pool and altered redox homeostasis compared to normal cells, making them more susceptible to artesunate-induced oxidative damage. And this has been shown through experimental models where ion chelation with deferoxamine can rescue cells from artesunate toxicity, confirming the ion-dependent mechanism. So at we actually have a team now that has been set up to basically look at a whole program of work on drug repurposing. And we are basically doing 2 clinical studies that I'll mention in a minute for colorectal cancer and also pre-cervical cancer. But as well as that, we are developing a framework for drug repurposing in Malaysia using WHO guideline standards. We're also doing a study on the barriers to drug repurposing in Malaysia and Southeast Asia. And we're looking at mechanism of action in nasopharyngeal cancer, colorectal cancer, as well as some microbiome studies of the effects of artesunate on microbiota and vaginome in our CIN 2 or 3 clinical trial. And we're also developing bioinformatics and machine learning features for drug repurposing development pipeline. And these are our PhD students and master students that are currently working with us at and obviously keeping Professor Ivy Chung very busy as well. So, in the clinical sphere, how we came to actually design the two clinical studies that we are currently doing in Malaysia was that back in 2015, Prof. Krishna published, before I joined him, a pilot phase 1 study, which was a randomized double-blind placebo-controlled trial of oral artesunate for colorectal cancer. And in this study, 23 patients were randomized to receive either 14 daily doses of artesunate, 200 milligrams once a day, or placebo. And results from 20 patients were analyzed as per protocol. And in that study, it was basically to show the tolerability and the safety of artesunate for 2 weeks before patients underwent surgery. And generally, it was well tolerated, although we did have 2 cases of grade 3 neutropenia in the artesunate group at day 14. And these were in patients who were under 50 kilos in body weight and also elderly. And when they had a bone marrow examination done, it showed that the features were actually consistent with a drug-induced neutropenia. So artesunate has a very short half-life of under 45 minutes. And basically the recovery rate after neutropenia is usually quite quick. So one of the patients actually recovered spontaneously, didn't require any further intervention after 24 hours. The second patient did have GCSF. But both of them went on to have surgery without any further complications. So that made us think a bit about how we design the study moving forward in phase 2. And we've actually set some weight limits where patients have to be 52 kilos as a minimum weight before entering the study, because we know that the neutropenia is dose dependent. And when we start to dose above 4 milligrams per kilogram, the risk of neutropenia basically increases. But apart from that, interestingly, the Ki-67 proliferation index was reduced in patients who received artesunate compared to placebo. And at 42 months, there was disease recurrence in 6 patients in the placebo group and 1 patient in the artesunate group. So obviously these are small numbers, but this is what led us to then design a phase 2 study, which again was a double-blind placebo-controlled trial of artesunate in stage 2 or 3 colorectal cancer. And to date, we have opened this study at in the UK at 7 centers in the UK, as well as 2 centers in Vietnam, and we have recruited 86 patients. And now we're opening this study in Malaysia with the aim of recruiting 114 patients so we can meet our final recruitment target. And the objective of this study is to determine the feasibility, safety, and preliminary anti-proliferation activity of oral artesunate in stage 2 or 3 colorectal cancer. And as I mentioned, patients will be receiving 200 milligrams once a day or matching placebo for 14 days prior to surgery. And then we're looking at recurrence-free survival 2 years post-surgery. And our secondary objectives include overall survival, drug-related toxicity, quality of life, surgical morbidity and mortality, and a biomarker and molecular profiling study. I won't go into the statistical power calculation, but if you'd like to know more, we can also share our protocol with you. And then these are the sites that we have opened up in Malaysia. I have basically been doing the clinical trial site initiation visits over the last 2 months across 11 different sites. And we're ready now to open the study to recruitment, and it has just opened to recruitment at this week. And then the second study that we're working on is looking at artesunate in pre-cervical cancer. So this is also based on some preliminary findings from the Johns Hopkins group back in 2020, which looked at intravaginal artesunate pessaries to treat CIN 2 or 3. And they showed that this treatment was safe and well tolerated. And about 70% of patients actually underwent histological regression, and 50% of those who underwent histological regression also experienced HPV clearance in this study. And what was interesting was that only the patients with ectocervical lesions experienced regression, not those with endocervical lesions, telling us that there probably was a topical effect. So in our study in Malaysia, we are actually using oral tablets because we feel that that's actually more acceptable to our Malaysian population. We're looking to determine the feasibility, safety, and preliminary anti-proliferation activity in CIN 2 or 3. And we'll basically be giving patients 3 cycles of oral artesunate, 200 milligrams once a day. And they basically have 14 days of treatment followed by a 1-week break to allow time for bone marrow recovery if there is any effects on the the bone marrow. And they'll do that for 3 cycles before undergoing a further biopsy and a LETS or a surgical procedure at day 90. And our primary outcome is to look at histological regression of CIN 2 or 3 on colposcopy and biopsy at day 90. We're also looking at HPV DNA clearance at day 90, comparison of the vaginome and gut microbiome, and adverse events and quality of life. And again, we have a recruitment target of about 28 patients in this study. I'm happy to share the protocol and the power calculation with you as well if you'd like to know more about that. And we're also taking the opportunity to do some translational research where we'll be looking at some cytokine parameters as well as vaginome and microbiome in these patients. And again, we've opened up at various sites across Malaysia and also at this week. And in the future, we are also in discussions about artesunate repurposing for polycystic ovarian syndrome, PCOS, inflammatory bowel disease, as I mentioned, renal nephropathy. And we're also interested to look at the HPV positive precancerous lesions such as anal intraepithelial neoplasia 2 or 3, vaginal or valvular intraepithelial neoplasia 2 or 3, and also oral intraepithelial neoplasia. And we're doing that in discussion with our colleagues in the Department of Dentistry. And in CMC Vellore, apart from AML, they're also working on myeloproliferative disorders and have a phase 1/2 study on MPN that will also be opening later this year. So I think I stop there, but just to say that we've had a very busy but productive few months formalizing IA data and with UM's participation also at Clinical Trial Connect, CRM conference back in May, and then also the EU Health Summit in May this year. And thank you very much to all our collaborators and colleagues on this journey so far. And we look to doing more together with and Malaysia in the coming few years. Thank you.
Thank you so much, Dr. Yolanda, for your great presentations. So right now we welcome questions. Please put your question on the chat or I think on the chat is the only platform to receive questions. There are a couple of questions already on the chat. Let me read some of them. First, thank you so much, Prof. Tengku Sarah, for your comments that she said it's great to see academics are studying drugs that will not be profitable to big pharma. So her question is, are there problems with funding due to this? So Dr. Yolanda, you want to comment on that?
Yes, thank you so much for that question. And yes, I think it is very challenging, and that's why it actually takes a long time to get these projects moving forward. But I think Malaysia is actually in a unique position because, as I mentioned, we do have 2 government-linked companies, Duo Pharma and Pharmaniaga, that are actually in the generic drug manufacturing space. So I think this is a real opportunity when we think about the global south. Like, for example, half the world's cancer patients actually living in the global south, and they don't have access to nivolumab or pembrolizumab or the immunotherapy that's costing 15 to 20,000 ringgit for 1 injection. But they do need affordable cancer drugs. So I think getting companies which are generic drug manufacturers in the global south to start thinking about going into this area of research is very, very important because the economies of scale can also make business sense. I think, we as academics, obviously, we also have to be practical. We also have to realize that industry does need to make a profit to be sustainable. But I think we need to push back against predatory pricing and also look at building partnerships where people like Duo Pharma, Pharmaniaga, etc., can participate with us in this and also as part of a national agenda for the benefit of patients.
Thank you so much, Dr. Yolanda, for your response. I hope that fulfills Prof. Tengku Sarah's comment. Okay, I want to follow up with another question from Prof. Tengku Sarah. She asked, what sort of tea should we drink to try to get some artemisinin? She's sorry to be controversial, but her question is that, is ivermectin still being studied for COVID-19? How you want to take that, Dr. Yolanda?
Yes, thank you so much. So essentially, I'm actually very interested in this as well because I think in Malaysia, there is a lot of research into the traditional medicines and the herbs space. And I think Malaysia is fascinating because I think just recently, last year as well, Ministry of Health did come up with a blueprint for research into traditional medicines and medicinal herbs. And I think this will be very nice in the future if we can look at how we can also integrate that together with drug repurposing, because I think we can actually share a lot of lessons and a lot of joint support in terms of how we actually design the clinical trials for testing these products. And of course, they have to be manufactured to the right standard. They have to be safe as we start off. But again, at the end of the day, we need clinical trials that are well designed, also practical and real world in terms of how we do this. So I think in the future, platform studies are potentially one of the ways to go if we can come together and look at what are the interesting medicinal herbs or traditional medicinal products that potentially could be put into good, well-designed platform studies. That would be one. In terms of drinking the tea, again, I'm very interested to go and speak to Chung to find out what they're actually doing with Qing Hao, etc., and if they have teas there as well. But I haven't had time to do that yet. But I think that is very interesting. But I don't have any recommendations for the tea per se. On the ivermectin side, essentially, I'm not an infectious disease specialist, but following the COVID-19 story, as far as I understand, there has been a lot of mixed outcomes of ivermectin in COVID-19. And to date, I haven't seen any trial data that actually supports changing practice to use ivermectin to treat COVID-19. But again, it could also be methodological issues with how the studies so far have actually been designed and implemented. And I think this is an area which is still open for discussion and debate. Yeah, I hope that helps. Yeah.
Yeah, actually, I hope you also answer Prof. David Oregon's question. He's asking what's the real deal about ivermectin. I think that hopefully that should answer Prof. David's question. Is it okay, Prof. David? I hope you can take that. Okay, there are another two questions coming up in the next 2 minutes. Maybe we can get it done. So there's a question about. Let me see where to wait. Where's the guy's name? Hey, wait. There's way back behind. Mm, I think I saw this one. There's asking about the how why you think you're choosing artesunate for the for as a candidate drug for your clinical trial.
Yeah.
Yeah, Sharifuddin, yeah, why you choose artesunate and what leads to the identification of artesunate as anti-cancer? And a very related question to that is just then yeah, also why you choose that for colorectal cancer specifically when there are so many other drugs that also safe for use and have potential anti-cancer activity. Any specific aspect that make artesunate highlighted? So I think they are related questions.
Yeah.
You want to do that?
No, thank you so much for that. So I think essentially it's also because artesunate is actually the first love of my PhD supervisor, who is also an infectious disease specialist who's worked a lot in malaria. So he had a lot of experience with artesunate, and when he started to see the preclinical data coming through for anti-cancer activity of artesunate, that actually piqued his interest, made him very interested, and that's how they did the phase 1 study at St. George's, which at the time was actually an unfunded study. So the surgeons and the infectious disease specialists just got together, put in their own funding to actually drive forward the research. So I think that's quite a interesting level of commitment. But at the same time, there are actually, as I said, over 300 different generic drugs which actually have potential anti-cancer activity. But I think looking at the therapeutic index, finding a drug which can target cancer with minimal side effects at the right dose for patients very important. So that's what we're hoping to do with the UMADT group as well over these next few years, is look at combination drugs as well, potentially how we might be able to use artesunate together with other generic drugs for the purposes of adjuvant treatment, for example, or neoadjuvant treatment. But I think all of these are fascinating questions, which we would love to go into more detail in a workshop where we can also share our learnings from how we looked at drug discovery and also what we're learning now through our partnership with Remedy for All, because there is a lot of, I guess, technological leapfrogging or a quick way up the ladder in a way, rather than having to, go around very laborious routes. If we also look at the bioinformatics space and pharmacology, networking, neural networking, etc. So I think there's a lot of stuff that we can do together, and we're not just confined to artisanate, but because a lot of the experience we have to date has been with artisanate, that's why we're pushing forward on this first. And I think we all need success stories. So we're hoping that if we can show some early successes or early wins with artisanate, that will make people more interested to look at other drugs as well.
Thanks so much. Yeah, so I hope that answers everyone's queries. Indeed, we are open if you have your favorite compound or you have good experience on certain compounds that you think is moving forward for drug repurposing, please reach out to us. And as Prof. Nungkusara mentioned, it's a funding problem. So if we get the right resources, right opportunity to bring everybody together, your favorite tea or favorite compound, maybe the next one in our clinical trials. So with that, I hope somebody's writing a comment. Let's wait for that comment to be finished. In case this is the next question. But it's 9 plus already, so I think really we have a good session today. I hope everybody can agree with that. Prashant, are you still writing a comment? I think you are still writing. I shall wait for you. Oh, is either okay. She just said thank you. Oh, I'm still writing. Do you mind, Dr. Yolanda, just maybe a few more seconds
No
in
problem.
case there's a question? Yes, for everyone's information, Dr. Yolanda comes a few times a year to UM and also to Malaysia. If you have anything that you think you want to discuss with us, just contact me and I will try to make that happen during one of our visits, the next visits. Okay, I sorry, Prashant, I have to call this off. Maybe you can write to me directly. Okay, with that, thank you so much, Dr. Yolanda, for sharing your journey, your projects, and also we hope we can open this for more collaboration with other academics in faculty medicine. All right, so I should close this webinar, and can I pass this back to the organizer? Thank you.